morning, ladies and gentlemen. Welcome to today's show on the Kasperick and Clemens Cognitive Coverage. My name is JP Kasperick, and I am joined alongside Audra Clemens. We have an intriguing topic today to discuss. Once we lose a sense, do your others improve? That's right. I am referring to cross-modal plasticity. Here's Audra with the latest.
It is commonly believed that once people lose a sense, their other senses improve after some time. Cross-modal plasticity is the brain's ability to reorganize the neural structure in order to have neurons function with multiple sensory systems. Due to cross-modal plasticity, when an injury or disease damages a sensory system, research has shown the brain does compensate for it, ultimately making other sensory systems stronger.
Although we know about cross-modal plasticity, relatively little research is known about the mechanisms of cross-modal plasticity and the process by which it occurs in humans. Based on experiments analyzing cross-modal plasticity in animals, researchers have formed several hypotheses. However, these experiments cannot be transferred to humans due to ethical issues.
Imagine waking up one day and your entire world is black. The sun that used to wake you up in the morning is no longer visible, and the image you would see of yourself in the mirror no longer there. According to the article Blindness, to approximately 8 million Americans, this is reality.
Blindness can occur to anyone at any age, but the brain's ability to adapt to the change of vision loss has a threshold between 14 to 16 years old. After the age of 16, the brain significantly decreases its ability to adapt to the change in neurons. The concept of neuroplasticity includes remodeling the organization of neurons and synapses in order to optimize the neuronal network function following developmental events, experiences, environmental factors, or brain damage.
More specifically, cross-modal plasticity is a type of neuroplasticity in which the brain reorganizes neurons to integrate the function of multiple sensory systems. Cross-modal plasticity is the process in which visual-related cortices are recruited to process sensory information from other perceptual sensory modalities, such as tactile and auditory systems in blind individuals.
Evidence for cross-modal plasticity is being shown in previous research, which has suggested enhanced blood oxygenation level-dependent or bold responses are present in the occipital lobe when participants were exposed to auditory or tactile stimuli as a result of early blindness. Luis, Sands, and Fine measured these bold responses for a variety of auditory and tactile tasks in sighted subjects, early blind subjects, and blind subjects using functional magnetic resonance imaging scanning, also known as fMRI. fMRI results showed the occipital lobe had a greater blood oxygen level dependency response throughout the auditory and tactile tasks in blind individuals than in sighted individuals. These results suggest evidence for cross-modal plasticity because neurons in the occipital lobe, once used for visual information, are now being used for auditory and tactile information, thus lighting up the fMRI, indicating more need for oxygen or more usage in the occipital cortex.
Research has shown that, on average, blind individuals become more skilled in their abilities to discriminate speech, detect echoes for guidance during locomotion, and engage in bimodal divided attention. Blind individuals are also better at discriminating between tactile texture than sighted individuals. Improvements in these areas seem to be related to perceptual functions, which are required in everyday life for the blind. On the other hand, blindness does not increase one's ability to detect pitch, rhythm, or loudness.
As of now, researchers are unaware about the method in which cross-modal plasticity occurs. Research has been done on animals, which has developed several theories about how cross-modal plasticity transpires in humans, as well as the mechanisms surrounding it. This includes changes in local connectivity, changes in subcortical connectivity, and changes in long-range cortical-cortical connectivity.
Local connectivity is the umbrella term that involves hypotheses concerned with cross-modal plasticity occurring near the damaged sensory system. Some hypotheses included in the local connectivity hypothesis are local sprouting, unmasking of silent synapses, and/or changes in the modulatory effects of lateral connections.
Local sprouting involves the growth of new, bushier axons with more dendrites than the previous damaged neurons. The features of the new axon allow for the formation of new neural connections in order to compensate for damaged or non-functioning neurons.
The new synapses are termed as being unmasked, which refers to those that were not in use prior to the vision loss, but are now needed in order to compensate for the vision loss. Finally, research concerning how the visual cortex adapts when receiving visual messages hypothesizes that there are changes in the modulatory effects in the lateral connections of the brain. Modulatory effects are produced through neuromodulators, a type of neurotransmitter, that diffuse through neurotissues to affect slow-acting receptors of many neurons, regulating areas of the lateral connections. Although there are lateral connections throughout the entire brain, this hypothesis is focused on the lateral connections in the visual cortex that receive and interpret the visual input.
Another theory to explain the occurrence of cross-modal plasticity are changes in subcortical connectivity. Research done in blind mole rats has shown evidence that portions of the brain, mainly functioning in the auditory process, are involved in visual functioning. More specifically, the inferior colliculus, the main auditory center in the body, was projecting to both the auditory and the visual parts of the thalamus to relay to get processed further. This research provides evidence that the brain may compensate for the loss of vision not only in the visual cortex, but also by establishing new connections in the subcortical region of the brain.
In each of these hypotheses, age plays an important role in the brain's ability to adapt to the change of vision. The importance of age during vision loss has always been difficult to determine, but through multiple studies, researchers have begun to determine that the critical region is until 16 years old.
In a study done by Sedato in 2002, early onset blind individuals and late onset blind individuals completed different braille tests to see if there was a difference in scores depending on the age of blindness. The results showed the early onset blind group was significantly better than that of the late onset blind group. This significant difference has led to further research and evidence determining that there is a threshold of 16 years that is critical for adapting to blindness.
Changes in the long-range cortical connectivity is another possible hypothesis behind the mechanism of cross-modal plasticity. This hypothesis has surfaced after research done in adult monkeys. Through research, it has been determined that there is a large pathway from the primary visual cortex to the area that represents peripheral vision. A great example of this hypothesis represented in humans is the ability to read lips, which is associated with the strengthening of the connectivity between the auditory and visual connections in the brain. Although this study focuses on the brain's ability to adapt through a speech impairment, it is still important to understand that cross-modal plasticity works bidirectional.
Although this has been shown at the behavioral and functional level, there is some controversy existing regarding the mechanisms that trigger these neuroplastic changes in the brain, mainly due to the difference in animals used and the time of sensory damage in the subjects.
To conclude today's news topic, it is apparent that our brains have the ability to adapt many different changes, including vision loss. Our brains adapt to visual impairment through cross-modal plasticity, which includes remodeling the organization of neurons and synapses in order to optimize the neural network function following developmental events, experiences, environmental factors, or brain damage.
Although our brains have the ability to adapt, there is a threshold of 14 to 16 years of age before the brain begins significantly decreasing its ability to adapt. Additional research is needed to determine whether the brain can catch up after the threshold. Well, that's it for now. Good night, lacrosse, and thank you for tuning in to the Kasperick and Clemens Cognitive Coverage.
Ladies and gentlemen, we have breaking news for you regarding cross-modal plasticity. We have just gotten word that a research experiment is being conducted in hopes of determining whether the threshold is concrete in brain adaptation. I'm being told that this experiment will use human cloned participants, which will allow them to look at the age threshold, the gender differences, and provide a stronger evidence for cross-modal plasticity.
The proposed research study consisted of 300 individuals. 50% were females, while the other 50% were males. There were 100 original participants who were followed and cloned twice from birth. There were 100 participants who were also followed and cloned at age 4 and tested at age 5. The other 100 participants were cloned and were blind at age 49. The sighted individuals were tested at age 5 and then again at age 50. The blind individuals went up to 4 years old and then got their optic nerves surgically removed, and then were tested at 5 years, and then again at 50 years. The third group of participants were cloned and blind at age 49, in which their optic nerves were also cut, and then tested a year later at 50 years old. When the blind cloned participants turned 5 years old, the pre-critical period testing was completed. To control for sight, the sighted individuals were blindfolded. Both the sighted individuals and the blind individuals were given a task with a tactile stimulus and an auditory stimulus. The task included a Braille reading test, as well as a tape recording of a voice reading a news article about common weather patterns throughout the United States, including heat, cold, and windy areas.
The researchers defined the independent variables as age, vision, the type of test, and gender, and the dependent variables as the test scores. Based off of previous literature, the researchers hypothesized that the cloned participants who were blind and tested at age 5 would do better than the sighted participants at age 5, and lifelong sighted participants would do better than those cloned and made blind at age 50. They also hypothesized that females would score higher than males. The results show that the hypotheses were supported. Results from this study showed evidence for cross-modal plasticity with a threshold age of 16 years. The participants that were cloned and blind and made blind at 50 performed significantly lower than those that completed the test at age 5, while cloned and blind. The sighted participants that were blindfolded at age 5 did worse than their blind counterparts, and participants at age 50 that were blindfolded did significantly better than their blind counterparts. There was also evidence that females adapted to the change of vision loss better than their male counterparts. Within the group of gender, there wasn't significant variability in the same age grouping. With the advancement of technology, future research should take exact time cloning into account when replacing this research. In addition, instead of administering different tests, future research can be done by surgically opening the brain on participants to analyze different cross-modal plasticity mechanisms. The surgery would allow researchers to remove different parts of the brain to see which parts directly affect plasticity and development. If participants are cloned during future research, the research will be able to compare the different brain structures and participants' ability to adapt to the vision loss.
The ethical concern of cloning humans was not taken into consideration during the study. Participants agreed to be cloned and went through extensive legal documentation to be included in this research. The participants were also paid for their time. With the exponentially growing scientific research and technology, researchers believe that this is the beginning of using the process of cloning humans in order to determine diseases, behaviors, and body functioning across populations. There is also no ethical concern about making participants blind during the different stages of their life. This ethical concern had to be ignored in order to fully understand and analyze how cross-modal plasticity occurs in blind and sighted humans.
This study confirmed that there is a critical period for cross-modal plasticity, as well as differences between gender. This research is important because although this study brought forward a lot of data, there were still some limitations. Only 2 age groups were measured. At age 5, individuals' brains were not fully developed.
The study also did not take in environmental influences into account. Although there were not significant differences within groups, the environment could still have played a role in the variability of results. We gave the participants 1 year to allow for development and the ability for cross-modal plasticity to occur.
Future studies could allow for more or less time and see if there is a difference between the learning and brain development length. At age 50, a person's brain is beginning to age and could have other cognitive issues aside from normal aging. None of the participants were measured during their prime cognitive ability, which is from ages 14 to 16.
Although technology has advanced, it still does not allow the clone to be created in exact time, requiring the normal time frame for normal human development. Therefore, the original participants and the cloned participants are 9 months apart in age. Based on our study being supported, there were no implications of this time and age difference within group results.